For the GST SH2 experiment, cells that contained the plasmid expressing the protein GST SH2 were grown by the teaching assistant, and the teaching assistant then used IPTG to induce those cells to make the protein. And they grew those cells with the IPTG in them for the recommended amount of time, and then spun them down. And this is the pellet that you now see for the experiment. Because you previously had experience seeing cells grow and reading them in the spectrophotometer, we do not expect you to do this part of the experiment because there's very little added value in learning how to grow cells a second time. So the first thing you want to do is lyse your cells. This is lysis buffer. So we're putting some lysis buffer, and the lysis buffer contains dithiothiotol. And we need to resuspend the cells in the lysis buffer, much in the same way that we've done previously, resuspending things, pipetting up and down. Now this lysis buffer contains dithiothiotol in addition to a buffer to give it a neutral pH, and it also contains detergent. So it contains pretty much the same type of things that you used in the plasmid prep experiment, the EDTA, the detergent, and in a moment, as soon as we're ready, I will put in the lysozyme. But first we need to get all the cells resuspended. Now that we've resuspended all the cells in that solution that contains tris and EDTA and triton, we're ready to add our lysozyme. This is 5 milligrams of lysozyme in this tube that we're adding to the resuspension mixture, and we're swirling the tube. And now we're allowing time for the lysozyme to chew away the peptidoglycan cell wall in the E. coli. This has to sit on ice for 30 minutes. Approximately 15 minutes into that 30-minute period, I'm actually going to be adding DNase. Now, in our aldolase experiment, we decided to skip the DNA step because previous work had shown us that the DNA from in this case, in the case of aldolase, the DNA from the chicken did not make a viscous solution, so we did not need to add DNase to get rid of it. But in this case, experiments have shown that when we break open the E. coli cells, it is a very viscous solution, and we were having trouble getting our beads that we added to it that contained the glutathione agarose beads. We were having trouble spinning them down because they were being held up in the solution because the solution was so thick with DNA. So we've actually added a step to this protocol to add DNase. So 15 minutes into the cell lysozyme mixture working, I'm going to add DNase, and I'm going to allow it to incubate for another 15 minutes. So still that same total 30 minutes. The DNase will chew up the E. coli genomic DNA, and then we will be ready to continue the experiment by centrifuging it at 10,000 RPM for 30 minutes. So the next scene you see will be after the DNase has been added, after the cells have been allowed to incubate, first 15 minutes with lysozyme, then 15 minutes with lysozyme DNase, and then after the lysos mixture has been spun at 10,000 RPM for 30 minutes, and we're going to be doing this in an SS34 tube. You have seen SS34 tubes before, so we're going to make sure that it's balanced in the SS34 tube with another tube with the same mass, and then we will be ready to continue the procedure after that point. We have now spun our sample in an SS34 tube in the centrifuge, and we now have a pellet consisting of the membrane. We have degraded genomic DNA in our supernatant along with our protein. Remember that we've added DNase to degrade a lot of that genomic DNA. We've taken a 1 ml sample of it and put it in a tube that is going to be our supernatant, and then we're going to pour it into a second tube. So we're getting rid of all the supernatant, keeping it in a separate tube. Then we want to add our glutathione agarose beads. So our glutathione agarose beads are agarose beads that have glutathione attached to them. And please recall that glutathione is the natural substrate for GST SH2. So our GST SH2 protein will bind to these glutathione agarose beads, and then the glutathione agarose beads will slowly settle to the bottom of the tube. So I'm going to let them settle and resuspend and settle and resuspend a couple minutes each time, followed by spinning in a tabletop centrifuge to get the beads to the bottom of the tube. So here we have addition of the beads. And then we're going to simply put the cap on it and allow those beads to settle, with occasionally inverting the tube to mix and letting them settle, and then spin to pellet out the beads. So the video will pick up after we have pelleted the beads down. Now we have centrifuged our supernatant with the glutathione agarose beads in it, and we see a pellet down here at the bottom. So we're going to carefully pour off our supernatant. You can also pipette it off, but if you do it carefully at once, not the back and not the back and forth stuff, but all at once, you have to commit. Then you can very carefully keep your pellet. The pellet's kind of loose, so you don't want to hold it upside down for a long period of time. Now, this is your flow through. So we're going to take a small sample, 100 microliter sample of our flow through to save to run on our SDS polyacrylamide gel, just in the same way we saved a sample of our supernatant. Now we want to wash the pellet off. We're trying to wash off any non-specific proteins. So we're going to add 10 mils of our wash buffer A. Put the cap back on and invert to mix. We're gently resuspending the pellet. And now we're going to re-centrifuge it so that we have a pellet again, and we're going to remove the wash buffer A. We're also going to save a sample of our wash A to run on the gel. So we're going to do the same thing with wash buffer A, and then we're going to do the exact same thing with wash buffer B. So when we resume the video, I will have pelleted with wash buffer A, removed the A, added the B, resuspended, pelleted with the B, and we will pick up from there. Okay, so now we have added our wash buffer B. We've gently resuspended in wash buffer B. Remember that wash buffer B compared to wash buffer A just has a little higher salt concentration, so that we're trying to get rid of any proteins that are not firmly bound to the beads. So now we're going to take a small sample of our wash buffer B to save, because we're going to run all of these samples on a gel. And then we're going to carefully pour off the supernatant into a tube that you can save, just in case. You never pour it down the sink until you're sure. And then we have our pellet right here. And to our pellet, we're going to add elution buffer. Now, the elution buffer has glutathione in it, and the glutathione is going to compete with the glutathione that's on the agarose beads. So our sample is going to, let me turn this to 1 ml. Our sample is going to come loose from the glutathione agarose beads and go into the solution. So we're adding 1 ml of the elution buffer to our pellet. and we're resuspending it. So we're gently resuspending the pellet. You can see some pellet material on the sides here in the resuspension. And so now we're going to centrifuge it very gently. We only have 1 mil in here. And after we centrifuge it, now our protein will be in solution. So next time, you do not want to discard the solution. You need to save the solution because that is your protein that's in the supernatant after we spin it down this next time. So now we have pelleted our glutathione agarose beads, and we are removing the sample from the top of the glutathione agarose beads. And I'm putting it into a tube. And then I'm going to take each of these tubes that are what I've collected a sample, and I'm going to take 100 microliters of the sample, mix it with 100 microliters of our 2x SDS loading dye so that the final concentration is 1x. And we're going to be loading about 20 microliters of that into each lane of a 12% SDS gel. So remember that previously we used a different percentage of gel, and you can use that same table to make whatever percentage of gel you want. The resolving gel can be 10%, 12%, 13%, whatever percent you want, and the upper stacking gel is always approximately 4%. So we'll pick up with loading. So just like we did previously with our SDS gel, we made the gel, the resolving layer, the stacking layer. We put it together the same way. I took the comb out, I filled it with buffer exactly like I did before, and I put a little bit of the dye in all the lanes so that you could see all the lanes. And now I'm loading my samples, my supernatant from the lysis, the flow through after adding the glutathione agarose beads, wash A, wash B, and finally the elution. So I'm just adding those to all of the wells one by one. Next to our elution fraction, I'm going to add markers, just in the same way that we had markers in the previous SDS gel. We have markers being added to this gel so that we can tell what size our eluted protein is. I'm then going to put the lid on, red to red, black to black. And we're going to turn it on. Running it at 100 volts, 150 volts, for approximately an hour until the dye front is at the very bottom of the gel. And then we will stop it. So we're going to go ahead and take it apart. We're going to pour off the buffer from the inside, open our gates, take our center assembly out, take our plate off, and you can see, well, I've run it till the dye is just at the bottom of the gel, and might be kind of hard to see, but there are separated markers right here. So here we have the stacking gel, here we have the resolving gel, and the markers have separated in the resolving gel. So now we're going to separate the plates, just like we did last time, and run a razor blade along the side to loosen it. We're not going to cut it in half this time because we're not doing an immunoblot. We're just staining the whole thing this time. We're going to put it in the stain. And we're going to rock it for about 45 minutes to an hour in stain. And then we'll take the stain off and begin the de-staining process, just first rinsing it a little bit with the de-stain to get most of it off, then 15 minutes, then maybe an hour, and then do a couple of overnights. And when you see the gel next, it will be stained with Kumasi blue.
To see the destaining, please watch the aldolase experiment on destaining.